Hello, everybody. I'd like to thank the organizers for allowing me to speak and also for giving me an excuse to visit New Orleans again, which is always fun. My name is Geoff Clark. I'm a professor in the Department of Pharmacology and Toxicology at the University of Louisville, and this project is a collaboration between myself and an assistant professor in the Cancer Center called Howard Doniger. And the technology is a first-in-class small molecule inhibitor of a cytokine called interleukin-6. So there are 16 cytokines of the interleukin family, and this is number 6 is perhaps the most important, or certainly one of the most important, because it's involved in a series of disease pathologies for which there's no good treatment or cure. One of those is uveitis, or dry eye disease. It's an inflammatory eye disease, which is caused by overproduction of interleukin-6 in the eye. It's estimated that the market requirement for this disease is around 500 million. Pancreatic cancer is another disease which is very, very heavily dependent on interleukin-6. So a lot of cancers are very inflammatory in nature, and pancreatic cancer is perhaps one of the most. We know pancreatic cancer has an absolute requirement for interleukin-6 because in the mouse models of pancreatic cancer, if you cross them with interleukin-6 knockout mice, they just won't form pancreatic cancer anymore. It seems to be an essential component of the development of the disease. And a third indication is cancer cachexia. This is the wasting syndrome that affects the majority of cancer patients. It makes them feel weak, helpless, hopeless, and just makes them want to give up. It's actually the listed cause of death on about a third of death certificates for cancer patients, and there's no effective treatment or cure. It appears that it's largely driven by an excess production of interleukin-6 by the tumor and the tumor stroma. So our solution to this is to target interleukin-6 with a small molecule. And just to give you a little bit of background biology in interleukin-6, because it's slightly more complicated than a lot of ligand receptor interactions. There's a classical signaling method over on the left, where interleukin-6 floating around in the blood can bind to an interleukin receptor on the cell surface. That's not an active complex in and of itself. The receptor interleukin-6 complex then binds to two molecules of GP130, and that forms the active signaling complex, which activates a growth signaling pathway called the JAK-STAT pathway, where the net readout for that is phosphorylation of a protein called STAT3, and that's helpful because there's a phosphoantibody against STAT3, so we have a biomarker for the activity status on the pathway. There's a second type of signaling that interleukin-6 can do, where the receptor itself is actually secreted into the blood, and so can form a complex that's soluble with the IL-6. It can float around the body and bind to any cell that expresses the GP130 molecules. So that means that even cells that don't have an interleukin-6 receptor can be modulated by interleukin-6, because IL-6 can bring its own receptor to the party if it has to. And then there's a third type where the complex is actually directly presented on the cell surface. And that means there can be a lot of microconcentration of the IL-6. So a little bit of IL-6 can go a long way when it's using the third mechanism. There's some competition in the space. There are actually 2 strange Russian antibodies which are not approved in this country for use against IL-6, which haven't been well tested. And there are 3 antibodies against the receptor, which have been approved mostly for rheumatoid arthritis or a rare disease called Castleman's disease. But there are no small molecules. So our solution to this was to come up with a small molecule inhibitor of interleukin-6. And we did this by in silico screening of drug libraries against the structure of interleukin-6. Buying in those chemicals, testing them through a semi-high throughput screening, biological screening assay, then doing about 8 rounds of medicinal chemistry to enhance the compounds and achieve composition of matter. We then have shown that the compounds bind directly to interleukin-6 with the KD in the low micromolar range. They can block the interaction of recombinant IL-6 with its receptor in pull-down assays, and they can specifically block molecular signaling in tumor cells. And this is just an example of this. This is a tumor cell line that expresses the interleukin-6 receptor. Over in the top left-hand corner, you can see a faint band. That's the activated level of STAT3 that's phosphorylated. It's quite low when the resting cell. If we sprinkle a little bit of interleukin-6 on it, you see that band gets much stronger. It's been activated. But in the presence of F4 and G1, which are early variants of our compound, we can completely suppress this activation going out to 4 hours. This is specific because if you look in the bottom 2 lines here, this is an analysis of what's going on with a different pathway called the MAP kinase pathway, and there's no change in the phosphorylation of that readout. So this is specific to STAT3. We've tested this compound in 2 in vivo models of our indication. The first one is in a uveitis model. There's a mouse model of uveitis. You actually inject the mice with an autoantigen to eye proteins, and they'll induce inflammation in the eye, and you can measure this. in the presence or absence of our drug, and we can see a statistically significant reduction in the clinical scores for inflammation, and also histopathological analysis when we introduce our compound by IP injection to the mice. And these studies were carried out by our collaborators in the Eye Institute. These are clinicians who see eye patients, so this is a model they use all the time, and they're quite good at scoring it. In the bottom figures, we actually then went to a second approach, was to use a topical application to the eye, which I think is the real advantage of a small molecule over an antibody. And again, we see a statistically significant reduction in inflammation in the eye. Over on the left is just the stats, and over on the right is just an example of what the eyes actually look like. And you can see the inflammation. You can actually see it going down visually without even scoring it, really. The second in vivo test we've looked at is in pancreatic cancer. This is a tumor model where we've injected immunocompromised mice with human pancreatic tumor cell line, then wait till the tumor reaches a measurable size, about 200 millimeters cubed, and then start treating with the drug and measuring the growth. And essentially, we can halve the growth of the tumor cells at the dose treated, which was 30 mg a kg, and we saw no toxicity at this level. Over on the right-hand side, we've looked at blood, urea, nitrogen, and NGAL. So we looked at the kidney and liver function and not seen any obvious change that's in a toxic region. And this isn't even the maximum tolerable dose, which we're still working up to. So just to conclude, we've developed a first-in-class small molecule inhibitor interleukin-6. It's active in vivo against uveitis and cancer models. It's got no apparent systematic toxicity at the therapeutic doses, and it also has a potential application to a broad range of other disease states. So cachexia is one, rheumatoid arthritis is another, the cytokine storm that was actually killing people from COVID-19, and then also influenza, actually, it should also be active against. And so at the moment, we're looking for partners to assist us with this and license the technology. And I'll stop there and take any questions.
So are you actually inhibiting the production of IL-6 or the interaction?
No, we're blocking its function. We're blocking it binding to the receptor so it can't work.
Gotcha. Thank you.
Yes.
Does it affect metastasis?
We haven't done the metastasis studies yet. My guess would be it probably does, but we haven't done that yet. The metastasis assays are a little bit more fiddly than the straight tumor ones, but that's something we're very interested in looking at.
It affects all kinds of cancers pretty much?
So it's pretty effective against inflammatory breast cancer, as you might imagine. We've tested it. I'm sorry?
TNBC.
We haven't looked at TNBC. The TNBC models, I don't think, are actually very good as models of the actual disease. Now, the lines you used actually don't look a lot like real TNBC, so we haven't put a lot of effort into that. It wouldn't surprise me. But I would expect it would have a much broader range than just pancreatic cancer. We've looked in lung, and it seems to work quite well in some lung cancer cell lines as well. But we don't have any in vivo data on that yet.
What's the therapeutic index of the compound?
At this point, it's hard for me to say because we've not really pushed it out. We were really just saying, does this work at all? And the answer seems to be yes. So that's really something we need to focus on a little bit more to give you a definitive answer of that. So I can't really answer that with any great degree of accuracy at the moment. Plus the fact the in vivo data was done on the older compounds. We've been through 2 new rounds of medchem synthesis since then, and we've got better binding and better signaling activity. So we're about to put those into the animals now to see how much better they are. And so I can't really answer that question.
Anyone else?
So 2 questions. One is, could you clarify the frequency of the dosage IP?
Well, once a day.
Once a day for the 10-day study that
Yes.
you did? Okay. And for improvement, what are you improvement against in this case? I mean, what are you looking for?
We're actually, so this compound will block the ability of a lot of tumor cell lines to proliferate in 3 dimensions. So that's one of our bioassays. And the other bioassay is signaling. What's the lowest dose we can use to inhibit the STAT3 phosphorylation? So that's the sort of readouts that we're using to try and make the compound better. Making the compound better is basically based on modeling and then on the previous results and then medicinal chemistry and a sort of iterative process.
Right. Thank you very much.
Thank you.